So this is truly our last glucose, our last sugar lecture that we will have in this course. Here we'll talk about storage mechanisms and controlling carbohydrate metabolism in general. This is chapter 18 from your textbook. This is going to be a 3-chunk lecture series, and this first chunk will start off by talking about glycogen, this short-term storage form of glucose, and specifically how we synthesize and degrade glycogen to store and release glucose respectively. The next chunk will talk about a process that we just mentioned here and there so far this semester called gluconeogenesis. If you break that word down, it means making new glucose from scratch or from nothing, and pretty much that's what this is. This is a salvage pathway to make glucose when we're running very low on glucose stores. And then we'll wrap up by talking about the regulation of carbohydrate metabolism from a whole body approach, not just in a single cell, but how the entire human body regulates glucose metabolism. So essentially what we're doing here is we're using this lecture to tie up the glucose story completely. We're going to wrap up talking about sugars by discussing how glucose is made into glycogen for short-term storage, how glucose is released from that short-term storage form, how it's released from glycogen, and how the metabolism of carbohydrates is regulated in general, not just cellularly, but from the whole organism perspective. So let's start off then talking about this molecule, glycogen. After any of us eats a good meal, by that we mean a meal that's high in carbohydrates, we typically have more glucose circulating in our bloodstream than we need to make ATP at the moment. Meals often mean excess for that moment. We don't need all of those molecules right away. We've already seen that high levels of ATP inhibit glycolysis, so if the cells have plenty of ATP on hand and you've just eaten a rich meal, it would be a waste to excrete all of that glucose. It would make a lot more sense to store it somehow. Of course, with ATP levels high, glycolysis is inhibited so that we don't convert that glucose into pyruvate unnecessarily, and so we'd like to store it. We'd like to store it for harder times to come. Animals have evolved to deal with harder times, and it's extremely rare for an animal to have a ready-made meal whenever they happen to be hungry. We've evolved to go long times and long stretches of time between meals. So this idea that as most humans, now hunger is a huge global problem, but most humans in this country do have meals when they want them, and of course my cats are always well fed, spoiled, rotten. We are walking around with bodies that have evolved to make do without. So we have this very efficient storage form for glucose. We can store glucose when blood glucose levels get high, and then release those glucose stores when blood glucose levels get low again. And in that way, we can keep glucose levels in the blood very, very constant and very stable. So we can absorb glucose out of the blood when it's high, when there's too much of it there, and release glucose back into the blood when glucose is running low, and always have a constant stream of ATP production. As we've said before, glucose is stored in animals. We mentioned this when we talked about starch, about amylose and amylopectin, and it's stored as a branched polymer of glucose, which we refer to as glycogen. Glycogen also goes by the less common name animal starch, and that's what it is. It looks very much like amylopectin. It's this branched molecule, and each of those circles you see on the screen, whether they're blue or they're red, is a single subunit of glucose. The only thing that distinguishes the red circles from the blue is that the red circles are endpoints. They are terminating glucose molecules at the end of this glycogen tree. So again, we talked about this a little bit as well, and I might be ringing some bells for some of you, but why a branched polymer? Why is glycogen synthesized in this way? The reason for glycogen being branched is the same reason that amylopectin is branched. We can release a single glucose molecule from a single branched end at any one time. The more branches we have, the more glucoses we can release in one shot, the more efficient the glucose release from glycogen will be. So evolution is pretty smart. This evolved structure for glycogen is the most efficient. With so many of those available glucose ends, remember in this diagram they're depicted in red, it is that many, many more glucose monomers that we can release all in one shot. If that chain of glucose were linear, like amylose, again, we can only release one molecule from an end at a time. We can easily see that in this case we could only release one glucose at a time, and that would be much less efficient. In fact, computer-based biologists who do experiments in silico, as it's called, have written programs to model the evolution of a glucose storage molecule, and they've programmed computers to evolve the most efficient glucose storage form possible. And those algorithms, those models, have come back with a prediction that the most efficient storage form for glucose would be a branched form, a branched molecule, where the average length of each chain in those branches were 13 glucose residues in length. That would be the most efficient, to have these branches grow no more than 13 glucoses. And any of you want to guess the average chain length of glucoses in a glycogen tree? You got it, 13. So evolution really has evolved the most efficient glucose storage form that can be conceived, even by computers that have modeled all possible scenarios. So that's glycogen. The question becomes, how do we break that glycogen down? How do we release those glucoses when blood glucose levels have fallen? Well, actually, we have two places where we can even put this molecule, and they differ on who they are storing glucose for. So we can store glycogen, we can store glucose as glycogen in the liver as well as in muscle cells. One of these is a selfish form and stores glucose only for itself, and the other is a selfless storage form and stores glucose on behalf of the rest of the body. It's the muscle cells that are selfish. Muscle cells store glucose as glycogen for themselves and themselves alone. As muscles become exerted, we talked about this with our challenge question with hunters, as muscle cells become exerted, they deplete through their ATP stores, they deplete their free glucose pools, and they use glycogen as a source of stored glucose to continue making ATP. So muscle cells that store glycogen are storing that glycogen only for themselves. Liver stores glucose as glycogen for the entire body. This is the selfless storage form. As the blood goes through and is filtered by the liver, there are also cells there that check the blood glucose levels in that blood as it travels through the liver. If those blood glucose levels are low, that's received as a signal that the liver should begin breaking down its glycogen stores and release that glucose into the blood to reestablish normal blood glucose levels. Once that glucose is released into the blood from the liver, it can circulate throughout the entire body and literally feed the rest of the cells in the body. Although we humans are now food rich, relatively speaking, we don't have to worry between meals. Still, this process that I'm describing occurs in your body between every single meal that you eat to keep your blood glucose levels constant. So between breakfast and lunch and dinner, and from dinner to the next breakfast, you're constantly either absorbing glucose from the blood and putting it into storage, or releasing glucose into the blood from those storage forms to keep blood glucose levels constant. In a healthy individual without diabetes or any other metabolic disorder, blood glucose levels are remarkably constant and vary very, very little throughout the course of a day. So the biochemical mechanism for releasing glucose from glycogen starts with a single reaction, and this reaction is shared in either liver or muscle forms of glycogen. Glucose is released from a glycogen chain in the form of glucose 1-phosphate, and hopefully you have some sense of what that name means already. That's simply glucose with a phosphate group on the first carbon, and you can see that here. So this diagram is meant to show you that glycogen is actually a molecule of glucoses, N glucoses large, where N is any number. If you add an inorganic phosphate ion, you can cleave off one of those glucoses, resulting in a glucose that has that inorganic phosphate on its first carbon, and the rest of the glycogen tree is made up of many, many glucoses of number N-1. That one missing glucose, well, you're looking at it right here. It's the glucose that was released. The enzyme that is responsible for catalyzing this reaction is called glycogen phosphorylase, which makes sense. If you think of what it means if I tell you that there is a hydrolase reaction, you probably immediately think that that's a reaction where some molecule has been cleaved by water. Hydrolase reactions or hydrolysis reactions are those where water is a cleaving agent. This is telling you that the enzyme is responsible for phosphorylase. Phosphorylasis, well, that's cleavage by phosphate ions. So already we know that this is an enzyme that cuts something using phosphate rather than water. And what it cuts, of course, is glycogen. So glycogen phosphorylase phospholysis molecules, specifically glycogen, in the presence of inorganic phosphate. And that's the inorganic phosphate that's doing the cleaving. Glucose 1-phosphate is then isomerized into glucose 6-phosphate, a form of glucose we're familiar with, and the enzyme that's responsible for catalyzing that reaction is called phosphoglucomutase. There it is again. Damn, chemists calling this a mutase reaction, because we're really not rearranging the glucose molecule itself. All we are doing is moving the phosphate group, and just like for phosphoglycerate mutase, when we moved the phosphate group and the phosphate group alone, that was a mutase reaction. The same is true here. So this is that reaction shown diagrammatically. The phosphate group is moved simply from carbon 1 to carbon 6. That's a mutase reaction, and so we are catalyzing that with phosphoglucomutase. I'm sure you know the answer to this question. We've seen glucose 6-phosphate before in our good old buddy, step 1 of glycolysis. Creating glucose 6-phosphate is the very, very first thing we do in glycolysis with the action of the enzyme hexokinase. I'm sure you all remember that well. It was not too long ago. And in muscle cells, where that storage of glucose was selfish to begin with, this glucose 6-phosphate slides right into glycolysis. It bypasses step 1 because it is already glucose 6-phosphate, and so this glucose 6-phosphate instantly becomes isomerized to fructose 6-phosphate, and then on to fructose 1,6-bisphosphate, and we know that story well. That's in muscle cells. Eventually, when the whole story is complete, we'll make lots of ATP from that glucose molecule via oxidative phosphorylation. Remember, muscle cells dipped into their glycogen stores because they were ATP depleted, and so this fixes that problem readily and problem solved. The liver, it's a little bit more complicated. The liver, remember, is releasing glucose for the blood, and it's storing glucose on behalf of the whole body. You might also remember that one of the points of phosphorylating glucose in glycolysis at step 1 was to lock it in the cell. So if the liver is going to release this glucose into the bloodstream, that phosphate on the 6th carbon, that phosphate that is blocking that glucose from leaving the cell, has to be removed. And so indeed, that's what has to happen. Once that phosphate is removed, that glucose goes out into the blood, circulates to the cells that need it, enters those cells, is rephosphorylated, and then enters glycolysis as we've described before. So that's all well and good, but glycogen phosphorylase as an enzyme has one serious limitation. It can't cleave glucose monomers off of branch points. In other words, if we look here, glycogen phosphorylase can remove this red glucose. It can remove this one, this one, this one, and this glucose here is a branch point glucose. It establishes this branch. And glycogen phosphorylase cannot remove that last glucose. If that were the case, then we could chew each of these branches down to their branch points and then be stuck, and there would be so much glucose left unyieldable. So if we look at this slightly more detailed view of glycogen, we can remove each of these glucose rings, but once we got down to this one right here, this is the branching glucose, we wouldn't be able to remove that. And we wouldn't be able to release any more of the glucose after it. So that would leave a lot of wasted, unusable glucose trapped in these large glycogen molecules, and we're not going to do that. We can't have that. So what the cells also have are what we call debranching enzymes, and debranching enzymes do what their names suggest. They remove the branch points and allow the breakdown of glycogen to be continued. So we can see that here. What actually happens is that 4 glucoses away from the branch, so you can see here's the branch, and then 1, 2, 3 more. So when there are 4 glucoses left on the branch, an entire block of 3 glucoses is moved in total to the end of a neighboring branch. So these glucoses that are shaded green are chopped off as a block of 3, and they're put onto the end of the molecule here, leaving only the branch point behind. And we see that it's been moved. Then one of these debranching enzymes comes and removes that single glucose that is making up this branch. With that single glucose gone, the branch is gone as well, and the cell can continue to release glucose monomers one by one via the action of glycogen phosphorylase. So this kind of two-step process, take a chunk of 3 glucoses off of the remaining branch, put it onto the end of some other branch, and then snip off that one last remaining branch glucose, allows glycogen phosphorylase to continue doing its normal thing and continue releasing glucose 1-phosphate as it goes down the glycogen tree. Glycogen is indeed a short-term storage form of glucose. It can be used much more quickly and effectively by muscle cells than other more longer-term energy stores can, such as fat, and this can be done anaerobically. Oxygen is not required to release glucose from glycogen, and as we already know, oxygen is not required for glycolysis. We can continue to perform glycolysis by making lactic acid and continue making ATP, and that's especially important for strained and depleted muscles that are being strained beyond their normal aerobic capacity. So this is a nice way to keep energy going. Burning fat is actually an aerobic process. I told you a few times already in this course, we do not have time to talk about beta oxidation, which is the biochemical process by which fats are broken down to acetyl-CoA and ATP made from them. But what I can tell you is that beta oxidation is a oxygen, heavily reliant on oxygen process, very, very aerobic. And so you may have heard this in non-scientific circles, but it is well known that any individual who is working out to burn fat should be doing low-intensity aerobic exertion, things like jogging slowly on a treadmill, stationary biking, and things like that. If there is oxygen available as those muscles go through their immediate ATP and glycogen stores, then that available oxygen will be used to burn fat and create acetyl-CoA from fat so that more energy can be provided to those muscle cells. Under high-intensity exertion, where cells become rapidly anaerobic, there's no oxygen around to burn fat, and so there the cells go through all of their glycogen stores instead. This would be high-speed cycling and other things that cause muscles to run through their ATP very quickly and become depleted for oxygen. So if there's no ability to burn fat when high-intensity exertion is occurring, then there better be a lot of glycogen around, otherwise cells are going to start to starve for energy. Many of you who either know of marathon runners or maybe are your marathon runners yourself, this is also true of long-distance cycling, the mecca of which is the Tour de France, of course. Marathon runners and professional cyclists are known to eat huge pasta-heavy dishes the afternoon or the night before their big events. And the idea of this is that pasta is, of course, starch. Starch is nothing more than packed glucose molecules, stored glucose from plants. These individuals, when they do this, are consuming far, far more glucose than they need at that moment. That glucose, of course, though, will never be wasted, and so that glucose is rapidly absorbed by cells, liver and muscle cells specifically, and turned into huge glycogen trees. Next morning, those individuals wake up, their cells are full of glycogen for this reason, and then as they undergo these high-intensity exertions and these anaerobic exertions, their cells don't run out of energy because they have all of these glycogen stores. So the idea of eating these huge pasta meals is to get and consume as much glucose as possible so that cells can make as much glycogen as possible before the event. So that's how you break glycogen down. That leads us to, well, how do you make glycogen? And it's pretty much the same process in reverse. Like so much else in metabolism, making glycogen is not biochemically the exact opposite of breaking glycogen down, but conceptually, it's almost the exact reverse of the same process. The problem is that making glycogen is endergonic. It has a positive delta G. As we take these random glucose molecules that are floating around in a cell and we try to order them into a very structured, tree-like, branched-like molecule, we're of course reducing randomness. Entropy is going down in that scenario. And whenever we decrease randomness that much, of course we're going to require energy to bribe the universe and let us do that. The energy for making glycogen actually comes from UTP, uracil triphosphate, one of the unique uses of UTP biochemically. So cells will hydrolyze UTP, and the energy used, or the energy released by hydrolyzing that UTP, is going to help us to activate our glucose molecule. So if we have excess glucose in the cell, the way that glucose stuck in the cell was that it was first phosphorylated upon entry. So that means we have high levels of glucose 6-phosphate around. That glucose 6-phosphate is isomerized to glucose 1-phosphate. And glucose 1-phosphate then reacts with this recently hydrolyzed UTP to give us uridine diphosphate. And that's what we see here, UDPG, uridine diphosphate glucose. Here's the uracil group, the ribose sugar, of course. The diphosphate refers to 2 phosphates in a row. And what's on the other end of that diphosphate chain? Here's our glucose molecule, what was just a moment ago glucose 1-phosphate. So UDP glucose, or UDPG, is our activated version of glucose that will now be able to go off and join a glycogen tree. I should point it out, it's actually pyrophosphate that we release from UTP. You're probably asking yourself, well, what do you mean pyrophosphate? Pyrophosphate is 2 phosphates. You told me this was UTP, triphosphate, and I see 2 phosphates in front of me. How did pyrophosphate leave this reaction when I see these 2 phosphates from UTP? You actually don't see these 2 phosphates from UTP. This phosphate is from uracil monophosphate. The other 2 phosphates were indeed lost. Where did this phosphate come from? Remember, the molecule that we're adding on to UDP is glucose 1-phosphate. So this is the phosphate group that glucose 1-phosphate brought on its own. And so we had a total of 4 phosphates involved in this reaction. 2 of them lost as pyrophosphate, 1 of them stuck to UTP, and the other came with glucose 1-phosphate. The enzyme responsible for making UDP glucose is UDP glucose phosphorylase. Makes some sense. It is a molecule that is breaking phosphate bonds as well as making UDP glucose. Incidentally, when those UTP levels become low because we are storing way too much glucose, again, this might be happening in a marathon runner or in a cyclist the night before the event, we can replenish those UTP levels by using ATP to give us diphosphates. And so ATP will phosphorylate UDP, and we make UTP for more glycogen synthesis, and we have ADP as a result. So you can make the argument that ATP is indirectly providing the energy for making glycogen because it's ATP that's keeping the UTP system charged. Now the big player in all of this, glycogen synthase, becomes involved. Remember, this isn't glycogen synthetase. That would mean that it uses ATP directly. This is glycogen synthase. All of the energy we need to make glycogen is already stored in UDP glucose. That's the activated form of glucose. So the energy between the UDP and the glucose is the high energy form, the high energy bond that we need to power this forward. So here is our UDP glucose, our uracil, our sugar, our diphosphate, and our glucose. And we're going to add that glucose itself to the end of a branch or growing chain of glycogen. That's done by simply breaking the bond between the second phosphate and the glucose. That bond breakage releases a lot of energy, and the energy release is coupled to the joining of this glucose monomer to the end of the glycogen tree. Remember, any sugar-sugar bond is a glycosidic bond. We covered that way back when we started this sugar story. And so a new glycosidic bond is made through the action of glycogen synthase. Glycogen synthase makes the vast majority of glycogen by simply adding glucoses one at a time, the way we just described, onto glycogen branches. But every once in a while, those branches grow too long. And when they do, branching enzyme comes, and branching enzyme creates branch points, just as debranching enzyme broke them. As a branch of glucose grows to be significantly larger than 13 glucoses, that will be a trigger that branching enzyme should come along and take about half that branch, chop it off in mass, chop it off as a chain of glucoses, and just stick that branch somewhere else on the glycogen tree. So you can see again, these shaded glucoses are removed as a block from the end of this branch and then used to establish a new branch somewhere else on the glycogen tree. So that's pretty much the story of short-term glucose storage. We started off by defining what glycogen is, taking a look at it. Glycogen is a branched polymer of stored glucose molecules, and it is how our cells store excess glucose for long-term storage. When blood glucose levels get low, we can tap into these glycogen stores and release glucose from the storage. We defined glycogen locations as two different places in the body, the liver and the muscle storing that glycogen only for itself, and the liver storing that glycogen for every other cell in the body. In both cases, glucose is released from a glycogen chain in the form of glucose 6-phosphate. That's a phosphorylasis reaction catalyzed by glycogen phosphorylase. Glucose 1-phosphate is then, in both liver and muscle cells, isomerized to glucose 6-phosphate. In the liver, that glucose needs to leave the cell, and so the phosphate is removed. That frees the glucose back to free glucose form, and that glucose leaves the cell and enters the bloodstream. In the muscle, that glucose 6-phosphate can instantly join the glycolysis pathway and be processed all the way to pyruvate. Then we switched gears and talked about making glycogen. To make glycogen, the enzyme glycogen synthase transfers a glucose monomer from the end of uracil diphosphate, where it's very active, a lot of energy released when you break that bond, to the end of an already existing glucose chain of glycogen, and that creates a glycosidic bond. Once that glycogen is made, it can then be released at any later time. The glucose can be released at any later time through the efforts of glycogen phosphorylase when blood glucose levels get low. So that's essentially the story, in so much as we need to know it for the storage of glucose long term, and this allows us then to go into other stories of glucose and metabolism in the second two chunks, namely making glucose from scratch when not only do we not have it in the bloodstream, but we also don't really have glycogen stores, and we got to somehow make glucose from scratch in order to keep cells alive. And as I said before, the whole body metabolism of glucose will come in chunk 3. Until then, as always, thanks for watching.